So hi everyone. So this is the start of this weekly's sort of club session. So today's session is sort of a continuation of the first weekly club that we had last week, which sort of tried to explore further sort of the macrophage subtypes that you find in idiopathic pulmonary fibrosis and how these could be either targeted or sort of used for immune modulation. So these were sort of some of the questions we tried to answer last week. And just as a background, I'm Alexis Garduno. I'm one of the scientists at Biotrain. So I'll be leading most of the weekly sessions. So you'll probably see me weekly, and I hope to see some of the users joining today join some of the next sessions that we'll be covering. So this week's session mainly will focus on looking at sort of the insights of lung macrophages from the Metareference tool with a specialized, yes, Son, you had a question? Okay. If you have any questions, you can always link them below in the Q&A or in the comment chat. If you can't hear me clearly or if you're having issues seeing the screen as well, just let me know. So as I was continuing sort of the webinar today is mainly focusing on sort of this meta reference, spatializing on sort of the database and also cross-referencing TALK2DATA and other types of modules within the Biotrain ecosystem, which is Biotrain Lens. So it's looking into how can you cross-link single-cell RNA-seq internal data, which you can perform cell type prediction, and cross-link that to Atlas integration from TALK2DATA and also spatial transcriptomics sort of insights into the same sort of lung macrophage etiology. So this would be sort of the main focus of today. Um, so welcome everyone. So going into a bit of an intro into the Biotrain ecosystem. So as I mentioned, the talk today will try to focus mainly on sort of harnessing the cell type prediction tool, the Metareference cell type prediction tool, and sort of integrating a new sort of feature update, which is the filtering of this sort of Metareference database. So this would be one that I will cover using our Browser X system. We'll then go towards how can you harness sort of these insights and output and integrate them into a larger sort of data set. So how can you create a customized atlas, which is able to give you even further insights and validation from your study? So this would use the TALK2DATA system. And from there, we'll try to cross-link how are these results or these different types of macrophages co-located on sort of lung tissue. And more specifically, how can we look at not only respiratory system diseases, but also apply it in other areas like lung cancer. And if we do see this cross-linking of these specific target genes. So this will be using our Biotrain Lens platform, harnessing sort of public data sets that you can use. Okay, so a bit of background on sort of last sessions, just in case some of the users in the call haven't joined. So last week, we tried to utilize the meta-reference-based cell typing tool to unveil specific macrophage subtypes in idiopathic pulmonary fibrosis. We noted that the monocyte-derived macrophages in this study played a huge role in sort of forming scar tissues in IPF and also having to do with the severity of patients in other respiratory system disorders. Following an example, which we did cover using Christina Moore's recent publication, we successfully applied the meta-reference tool to re-annotate these macrophage subtypes in IPF. So this was sort of a tool that we covered extensively in last week's session. And if anyone from the call do want to sort of recap that session to be able to then build from this new talk, we do have that sort of recording accessible through our sort of database system. Okay, so just let us know if you need any links. Okay, so just to recap a bit of the meta reference, so you can sort of place what this means for any new users who join the call. So the meta reference cell typing tool, basically it's a library of nearly 1,400 human public data sets, which adopt specific gene markers for different cell types and cell subtypes. By comparing your data with the vast collection in this meta reference tool, repository, we can identify specific similarities that make sort of informed predictions or that help you sort of predict the final annotation of your specific cluster or cell subtype. So basically, just think of it as your data set is a book, which we are trying to cross-link and reference and sort of compare these different gene markers across other books from our whole repository in TALK2DATA. So this is what Metareference cell typing tool is sort of built on. It's based on all of the curated data that we have, and we harness that power of all those millions of cells to be able to then predict and help you or support you in assigning a final annotation for your cluster. Okay. So a bit of results that will help you build in what we're going to see today. So in the last session, we were able to use the meta-reference based cell typing tool, and we were able to predict these 3 macrophage subtypes. So we identified that these IPF patients, and not only IPF, we were able to cross-link that with other studies relating to the severity of COVID-19 and also other allergy-related diseases, for example, asthma. So some of the 3 subtypes that we'll be sort of looking into even further today is the SPP1 high macrophage, the FCN1 high macrophage, and the high enrichment of this FAP4 macrophage. So these similar expression patterns of SPP1 and FAP4 macrophages across IPF, allergy and COVID-19 are sort of the hallmark of this sort of lung inflammation and potential treatment targeting that we want to sort of dive even further with these new updates in the system. Okay. This week, just to recap a bit more, so we'll be going into the meta-reference cell typing tool, and we'll try to predict using specific lung-related studies. So in the previous session, we mainly used the meta-reference tool, but we used all of the studies used in the prediction model as sort of its training data set. But with this new update, we'll be able to actually select very specific data sets relating to the tissue of interest. So in this case, all lung tissue related data sets. And we'll also be able to add specific sort of filtering based on the conditions that we're trying to analyze even further. So we'll be sort of adding respiratory system disorders and also other related sort of cancer disorders in the lung. So these would be some of the additional features we'll be covering and how can this further add other pictures into the analysis. Okay. So the start of the demonstration today, so I'll be mainly demonstrating these functions through our platform. So as you know, these are sort of the 3 platforms that we'll highlight today. So if we go directly into our bBrowser X system, we can then pull on our data set that we'll be covering today. So this was the IPF data set that we were analyzing last week. So we'll open up this study. And this is where we'll try to apply the cell typing meta reference tool, but with this new filtering capability. So how can we further distinguish all the data sets and make them specifically trained on the case study from the sort of analysis that we're mainly sort of focusing on? So the first step, as I mentioned last week, just for anyone who is here, you have to make sure that you run the Louvain clustering. So this network-based sort of methodology that is able to sort of rank the different clusters based on the expression pattern. So you'll have to apply this specific methodology first. You have to make sure that whatever data set you're mainly applying it on, that is batch corrected if you have different samples or different batches that have been uploaded into one study. So just make sure that you do correct using Harmony. That way it's able to find the resolution and be more discrete in the actual clusters. So this would be sort of the first step. The resolution that we normally recommend is 1. You can decide what resolution you want to apply. Normally 1, which is the highest sort of level, would mean that you have more number of clusters, which allows you to even distinguish further cell subtype levels of the data. So you can think of it, low would be less clusters, but more cells, so major cell type category. And then as you want to start identifying further cell subtypes, you can increase sort of this resolution. So that would be the first step, running the Louvain clustering. So once that is run, you can then move forward to applying the cell type prediction meta-reference filtering. So I'll be going into that. Normally, it takes a few seconds to run the Louvain clustering, especially depending on the number of cells from your study, but it shouldn't take too long to run that analysis. Okay. So now that we have our sort of identified clusters, we can then move forward to the cell type prediction. So previously in the last session, I showed you that you can use the meta-reference tool and you can apply it on your identified sort of cluster resolution. But we now have this new feature that is the main focus of today. How can we sort of specify what tissue, what conditions we want to train the model on? So when you select filtering, you'll be able to sort of go into the specific tissue that you're focusing on. So in this case, since we're mainly working on sort of the respiratory system, we can then make sure to select sort of all the categories within the respiratory system. So when you sort of select that category, you'll see that you can even be more specific as to what areas in the lung you're mainly focusing on. So you can select studies which just have sort of the category lung, or you can be more specific into your location in the lung as well. So that would be sort of the first filtering. The second filtering that we'll try to apply is in relation to the actual condition and disease that we're focusing on in this study. So in order to be able to make the annotations more discrete in the prediction model, we have to make sure that we select the specific sort of disorders that are from the actual data set. So we'll select the respiratory system disorder category under condition, and we'll also select this respiratory system cancer or benign tumor. Since we are trying to see if there's any indication where this SPP1 cross-linking with FAP-P4 has also sort of a role or a functional role in cancer as well, relating to the lung. So once these are selected, you can make sure that you select the correct sort of resolution that you ran the Louvain clustering for, and you can then just predict the result. Okay, so this will make sure that it's only including data sets that are sort of similar to your specific study in this case. Okay, so once this sort of analysis is run, you'll be able to explore the supporting data sets even further. So this was part of it that we covered in last week. So how can we see an overview of the different clusters and what is the percentage that is being assigned for each cell type annotation based on the model or the meta-reference repository. So this is something that you can directly go into the clusters of interest. So in this case, since we are focusing on further identifying sort of these lung macrophage functional cross-linking sort of target genes, we'll go into the clusters that have these specific annotations. So in cluster 7, you can directly identify that the system or the meta-reference repository directly assigned a percentage of 64% that are sort of positive for this macrophage annotation. So if you want to then explore this further, or if you want to further identify, let's say, the specific target genes or gene symbols for that cluster, you can then further sort of look into the actual details in the genes. So how are these genes ranked as opposed to the other clusters, and how can you further define these markers? So we can directly see that in cluster 7, there is a high SPP1 sort of expression. So if we want to then further analyze this, I'll go into the major cell type category first, and I'll explore this analysis. So we can see here that these sort of data sets are used for the model. So since we specifically selected data sets only in the lung and only in respiratory system disorders or in lung cancer, we can directly see how these authors specifically sort of identified these results. So in sort of the cluster 7, we can see that there is a high enrichment where this other authors define this SPP1 high macrophage. And then we can go see if there's any other clusters that are giving any sort of cross-linking relationship with FAP4, which was another sort of target that was highly enriched from our previous session. So if we then go into the overview and we try to go into cluster 11, which was another one that had this defined macrophage group, I'll go into the major cell type annotation and I'll try to explore this cluster even further. So we can see here, if I go into these data sets, I'm also seeing this FAP4 high macrophage. So we can then validate that even with the filtering, you're still seeing sort of this FAP4 macrophages, which are benefited from the reference library, and that also sort of are linked to our previous 3 research groups that we sort of analyzed last week. So what can this tell us further? So the recurrent occurrence of this SPP1 and these FAP4 enriched macrophages has to sort of do or has some functional link to lung tissue samples. So we can also sort of try to analyze even further how can this occurrence of these markers in these macrophages across these studies strengthen our hypothesis. So are these 2 clusters closely linked to just lung-related diseases? Or can there be an observation which aligns with this co-regulation across multiple lung data sets? So not only lung-related, but also cancer data sets, since it was used in the filtering of the meta-reference prediction tool. Okay. So to further sort of analyze this co-regulation and to further see if there's any relationship with targeting SPP1 and how can this reduce some pro-fibrotic activity in these macrophages, we can then harness what is sort of all the repository and TALK2DATA. So how can we move from a 1-level data set and then integrate that into sort of an atlas integration. So multiple data sets relating to the same disease and sort of looking if there is any cross-linking across these 2 markers. And if we only see this cross-link in just respiratory diseases or lung-related diseases, or can we also move into sort of the lung cancer space as well? So for that sort of part of the session, we'll go into sort of the TALK2DATA module. So I'll bring in sort of our ecosystem page and I'll go into the TALK2DATA system. Okay, so within the TALK2DATA system, you'll see that the sort of repository of all these studies are built in based on species. So you have to make sure that you select the correct species where you want to sort of integrate these studies from. So if it's specific to mouse experimental models that you want to build an atlas from, or if you want to sort of build an atlas from a specific technology, just make sure that you select sort of what data or what technology you'll be building the atlas from. So since in this case, our IPF data set is mainly in human and it's just single cell RNA-seq, we'll try to then build in a specific atlas integration relating to human studies in the single cell RNA-seq database. So under studies, this is where you'll be able to then find all of the data sets curated under that specific category. And we'd also have this advanced filtering tool. So in the filter tool, you'll be able to sort of open up our standardized ontology system. So this is how we're able to really normalize and curate all the data sets so you can actually integrate them and sort of harness the power of sort of multiple studies and more number of cells as well that you'll be using to cross-link this information or to validate the cross-linking of these targets. So when you're in the filtering, we'll then go into the same filters we applied for the cell meta-reference cell typing tool. So in tissue, you have to make sure that you're only going to select specific tissue relating to the lung, so the respiratory system. So I'm just going to select the respiratory system here. And if it's hard to find it in the dropdown, you can also search for it as well so that it's easier to sort of select all the areas in the respiratory system. So once that filter is added, you can then move forward to the actual condition. So in this case, I want to be able to sort of validate if these targets or cross-linking have a functional sort of role where it has to do with mitigating inflammation, or if it's sort of the macrophages are sort of stabilizing the lung tissue in itself. So I'll be able to go into condition and then select any sort of conditions relating to the respiratory system. So they'll normally be all the way at the end. So once I select the respiratory system disorder, I can then move forward to the actual cell type that I'm targeting. So this is where, again, if it's easier for you, instead of going through all of the cell type annotations, you can directly search and then select all of the macrophage cells within these data sets and within this tissue. So I have my three filterings, which you can see here, tissue, condition, and cell type. I can then search through the system to be able to then pull in all of these different cells across these studies. Okay. Once these studies have been located and it gives you sort of the number of cells that you'll be able to sort of use, you can then move forward into creating your custom data set from here. So this might take a few seconds just until it searches for all the data sets in the system. So if it does take a while, I do have one which is ready just in case. Okay, so this might be taking a while to find all the studies. So there is a way that I can bring that information. So in the custom data set, you'll see that I have had created this respiratory disorder within the lung tissue and this macrophage beforehand. So it was able to find from the database 44 studies and 420,000 cells that are just macrophage specific. So no other cells. I'm only focusing on these macrophages. So if we explore this atlas even further, you can then see how this integrated atlas has integrated all of the 44 studies, and you can then go into the information relating to the condition. So it definitely has a higher power because you have many cells relating to also normal sort of disease and the respiratory system disorder. So we can actually compare if there's any differences where the macrophages are sort of stabilizing the lung tissue across just normal donor samples, or if there is a difference in relation to the severity of the disease itself. So to sort of show you some of the results, if we use the composition chart, we can then pull in information relating to sort of target analysis. So you can input your list of targets and you can sort of build in what labels or what metadata sort of clinical analysis you want to perform. So how you can see the proportional differences across the cells and what other component analysis you can build from there. So just to sort of save some time, since it is a lot of functionality, we can then go into some of the results that we're sort of interested in exploring further from the atlas. So if we take what the results showed from the IPF data set, so the single study that we were analyzing from Christina Morris, we could see that SPP1 and FAP4 do have some distinct expression patterns, especially when it comes to normal healthy donor samples compared to sort of IPF samples. So we can see that in this case for SPP1, we do see sort of an upregulation in IPF, whereas for FAP4, there was sort of a higher enrichment specifically only in healthy donor samples. So this was sort of results that we gathered from one individual study. But if we then combine, so from our lung macrophage atlas that I showed you how to generate, we can query those two genes and try to map if there's any differences that I'm seeing from like a larger macrophage atlas in the lung. So we can see here that we're still seeing this similarity in the normal healthy donor. So we can see that FAP4 is specifically a target which is enriched in healthy donor control samples, whereas it has a low enrichment in respiratory system. And if we look at SPP1, we can see that there is a role mainly in the respiratory system disorder. So it can give us a bit more detail on how can this sort of SPP1 marker mitigate this inflammation in sort of lung tissue, but also the disease that we're focusing on. Okay. So this is just, again, recapping on that information. So if I go back into my sort of atlas, there is another part how you can take this even further. So if you're trying to then sort of analyze, okay, what is this insights giving me and how can I then analyze further? How do these cross-linking of these two targets express across different areas in the lung? So how can we move from single cell RNA-seq into spatial transcriptomics analysis and be able to then reference, okay, what area in the lung, and is it only specific to respiratory system disorders or also lung cancer as well? So in order to be able to analyze this increase of SPP1 and the decrease of FAP4 relating to the pathological environment and how can it promote either fibrosis or suppress the immune system, we can then harness the bioterrain lens platform. So within the TALK2DATA system itself, if you're interested in sort of applying these insights and if you want to cross-link them to the actual pathological environment, you can then go into different spatial transcriptomics databases to be able to locate specific studies that are relating to either the tissue of focus or the condition as well. So each of the databases, whether it's spatial, single-cell, RNA-seq, will always allow you to filter what studies you're mainly focusing on. So in order to do that, let's say we then go into this specific lung tissue data set relating to non-small lung cancer. And we can then query some of the markers that we're really interested in seeing how can these contribute to the pathological environment. So if we look into the SPP1 specifically, and I zoom out, we can see that SPP1 is mainly concentrated on very specific regions of the tissue. And this can sort of further give us insights if it's contributing to specific regions in the tumor site or lesion sites if it's another type of respiratory system disorder. So you can see that there's a huge transcript. So if I zoom in, one way to sort of interpret this, so these segments are sort of the segmentation tool that is segmenting each cell within the tissue. So this is single cell resolution spatial imaging analysis, and each individual cell is within each of the segments. So you'll see that when we query a specific gene, it's the actual transcript information of that gene target. So some cells might have a higher enrichment of this sort of SPP1 sort of transcript. So you'll see that especially for some tumor cells or some lesion areas or tumor site, primary tumor areas, you do see that this SPP1 marker is highly sort of focused or expressed, and it's expressing very sort of numerous type of transcripts within one cell, for example. So this is a way to interpret this, okay? And if we're trying to analyze, okay, how can we then see what is the relationship with FAP4? So if I then click on FAP4, we can see that FAP4 is more like normally like distributed across the entire tissue. So it's not really sort of focusing on a very specific area or in a sort of tumor site, and it's not really driving that inflammation in this case. So it's more sort of a more stable marker, whereas SPP1 is really driving sort of specific cells into those specific sites, which are either inflamed or sort of have a specific role in the type of tumor in this case for this lung cancer data set. So how can we sort of analyze these clusters further? So as you know, if we're mainly targeting macrophages, SPP1 as a marker is not the only target that is expressed in macrophages. So if we want to validate sort of the base sort of spatial distribution of SPP1 and how can it create sort of immunosuppressive niches in the tissue within the tumor microenvironment, we can query other specific genes in relation to this. So the first thing that we'll do is select the niche category so we can see all of the cell types that are sort of curated within the tissue, and we'll remove the FAP4. So what we want to see if there's any sort of functional role for these macrophages and how can these targets drive this immunosuppressive environment. So another target that is normally found in macrophage is the IQA. So that's another important target. 1QA. Okay, so if we have these markers, we can go into the sort of niche category and we can try to sort of ensure that these cells are specifically macrophages. So in order to do that, we can query this specific sort of gene. So if I zoom out, I can see that these macrophages, which are the ones in green, are all sort of expressing this SC1QA. So this is sort of an extra validation where we can see that this marker is specifically sort of mainly in the macrophage area. And similarly, if we go into SPP1, you can see that many of these macrophages are also sort of expressing this target in the tissue. So how can we further sort of analyze this disruption within the tumor growth, or how can we see the potential role that it can have in tumor progression? So another marker, which is mainly known as an antigen presentation sort of marker that plays an active role in immune surveillance, is the HLA-DQ A1. So if we want to see then if there's any role as well relating to this type of marker, we can then query that marker as well, and we can try to see if the expression is also located. So we can see that again, we see these macrophages that are mainly sort of expressing this HLA-DQA1 sort of target, and we can see the transcript information is mainly enriched within all of these macrophages in this tumor site region. So if you want to further sort of apply this with the actual sampling site, you do have other information that you can gather as well. So you can see if there's any relation to the actual sampling site in the tumor with these markers that we're analyzing currently. And basically, when you see this expression pattern, this suggests that the SPP1 high macrophage sort of subtype plays a huge role in suppressing the immune sort of response against sort of this lung cancer sample. And you can also see that the lower expression of HLA-DQ compared to SPP1 regions implies that these cells may help tumors invade immune detection. So you can see these different roles of these targets and how they cross-link in the context of the sort of tumor environment. So this is sort of how you can harness as well spatial imaging analysis to be able to replicate what you're seeing in the single cell RNA-seq space. Okay, so if I go back into sort of our slides, some of the summary that we can gather from this. So we directly observed and explained how SPP1 is highly expressed in disease macrophages, which indicate this role in the immune suppression rather than protecting the immune system. So it's driving more of an immunosuppressive state. And using the filter feature in the Metareference tool does allow you to even further predict very specific and spatialized annotations that are relevant to your research. So it makes it specific to the tissue or the location of the sampling site that you're mainly focusing on. We did see that the SPP1 and FAP4 expression patterns in macrophages are stable across lung-related diseases. So we could see this as well in the spatial imaging analysis using the lung related study. And we also found that the SPP1 macrophages may suppress the immune system. So it's rather than protection, it's more of a aiding this tumor progression in the sample. Okay, so we'll now go into sort of the Q&A. If there's any further questions that any sort of researchers in the call want to ask, we can ask these now. Okay. So here I'm going through some of the questions that are being asked. So do you have a white paper for this method? Can you share the link? So currently we are sort of developing the last changes on this publication that will sort of include more on the methodology from the Metareference cell typing tool. So we'll be sort of able to share it very soon. So it's in the process of being sort of submitted. Okay, can I use my in-house studies as a reference database? And this is a question from Olivia. So yes, the further sort of direction of the Metareference cell typing tool will be not only to filter based on our prediction model, but you can also sort of I guess, filter based on data sets that you have from bBrowserX as well. So that will be sort of a further direction that we're currently developing in the cell typing tool. One thing that I would recommend if you are currently going to use the cell type tool, there is other methods that might be useful. So if you have, for example, very specific gene sets that you want to apply in the model, so these are gene set lists for specific cell types, you can also use this gene set prediction tool as well. So we have other types of models that might be more specific to your research needs or your study in this case. Okay. So do we have any other questions from the team or from other researchers that have joined the session today? So if no one has any other questions, or if you do have very specific questions that you don't want to share, you can always link them through our support at biotrain.com, and we'll be able to connect with you directly if you have a very specific use case where you need our support on in terms of how you can harness this meta-reference cell typing tool. So for now, since we are sort of at time, this sort of concludes our sort of session for today, so our weekly talk. And as a reminder, this is a weekly sort of exploration club type of seminar. So we'll be holding a different type of seminar on a different topic next week, and we'll share sort of the topic for that session. So thank you everyone for joining. It's always great to see a lot of interest in this field, and I look forward to seeing all the users in the next session. Okay. So thank you, everyone.